Bacteria are everywhere. They pervade many aspects of our lives, in the air, in food, and even in our bodies.
I need your help to perform an experiment.
What did you have in mind?
Well, these culture dishes have the right mixture of moisture and nutrients to grow bacteria. What I want to do is cultivate some bacteria from your body. So with this sterile cotton swab, I'll take the bacteria from your forehead, and from between your fingers, now open your mouth.
What for?
I want to take some of the bacteria on your gums.
Are you saying I have bad breath?
No, I just want to see if there's bacteria present in your mouth. Now with this fourth dish, I'm following the same procedure, but I'm not touching you with the swab. Why am I doing this?
The fourth dish is an experimental control. It won't have any of the bacteria from my skin, and that way we can be certain that whatever grows on the other three came from me.
This incubator will provide the ideal temperature for whatever's on these plates to multiply. What do you think will happen in each dish? This is the control. Any bacteria here probably settled from the air. Now, from between your fingers, we have lots of different bacteria. From your forehead, we also have large bacterial cultures. And from your mouth, we've grown a whole bunch of bacteria.
Bacteria aren't always harmful. For instance, yogurt contains active bacteria with milk. The bacteria do us a service by pre-digesting the milk, making the proteins more useful to us.
Bacteria are also present in large quantities in our intestines, where they aid in digestion by breaking down compounds.
A wound or scrape can introduce bacteria into the body, causing infection. The immune system identifies these foreign cells and destroys them, but occasionally it needs a little help in the form of antibiotics. Antibiotics are chemical compounds that are toxic to bacteria. For an antibiotic to be useful in treating infection, it must be toxic to the bacteria, but not to the patient.
Since the discovery of penicillin in 1928, there have been hundreds of compounds discovered and synthesized that kill bacteria. One such group of compounds is called sulfa drugs. Sulfa drugs are toxic to bacteria because they are very similar in molecular shape to an important compound called para-aminobenzoic acid, or PABA. Bacteria use PABA to manufacture folic acid, which is vital to their reproduction. Folic acid is assembled in several steps. One of these steps involves PABA. Sulfa drugs are structurally similar to PABA, so that they are substituted during this process. This disrupts the series of reactions and prevents folic acid from being made. Without folic acid, new cells cannot be made and the bacteria die. Human cells can't manufacture folic acid, but rather acquire it from foods that already contain it. So the sulfa drugs are non-toxic to people. This is one of many examples in biology where the shape of molecules is very important in determining their chemistry.